think we're going to go ahead and get started. So good morning, good afternoon, good evening, everyone, depending on your location. My name is Vaki Odwaran, a co-chair of the H3Africa's Microbiome Task Force. Thank you for joining us for what I believe is going to be an insightful webinar by Professor Clement Adebamowo. Just to briefly introduce the very well esteemed researcher in his field with wide spanning affiliations, Prof. Adebamowo is a professor of epidemiology and public health, a member of the program in oncology and the Center for Vaccine Development and Global Health, and the Center for Research in Aging at the University of Maryland School of Medicine. He is the Associate Director of the Population Science Program and Director, Global Health Cancer Research at the Marlin and Stewart Greenbaum Comprehensive Cancer Center in Baltimore, Maryland. He is also a volunteer research scientist at the Institute of Human Virology in Nigeria and the Center for Bioethics and Research in Nigeria as well. Professor Adebamowo conducts research and implements training in research ethics, epidemiology, and genomics of breast and cervical cancer and other non-communicable diseases. He's the PI of a number of human hereditary health in Africa projects, including the African Collaborative Center for Microbiome and Genomics Research at the Institute of Human Virology in Abuja, Nigeria, the African and Indigenous projects. Thank you, Prof. Adebamowo, for taking the time out to share some of your work with us today. Just for a little bit of a housekeeping, there will be a question and answer session at the end of the talk, so please use the Q&A feature for any questions and talk-related comments or thoughts you may have. Thank you. Thank you again, Prof. Please go ahead. You're muted, sir.
Can you hear me now?
Yes, I can now. Thank you.
Okay, thank you. So good morning again. Today I'll be sharing our research into the vaginal microenvironment and the role that it plays in the risk of persistent high-risk HPV infection and cervical cancer. So as been mentioned, I work both in the US and in Nigeria, where I focus on cervical and breast cancers, among other things. And the research I'm presenting today was conducted at our H3Africa funded collaborative center in Institute of Human Virology, Abuja, Nigeria. And we focus on cancers because they have become an important public health problem for many African countries, including Nigeria. And the commonest cancers we see are those of the breast, followed by the prostate and the cervix. Today, I'll be discussing research around cervical cancer. Now, cervical cancer is caused by persistent high-risk HPV infection that has become transformative. But that is not the only infectious disease-associated cancer in Nigeria. In fact, many low and middle-income countries suffer a disproportionate burden of infections-associated cancer. On this slide, you can see the different types of infections that are associated with cancers and the burden that they impose on the population. In Nigeria, 23.8% of all cancers that we see are associated with infections, and the human papilloma virus is by far the most burdensome. So papilloma viruses are one of the oldest and largest family of viruses. And I think with the COVID pandemic, many of us are now paying more attention to viruses, but they've been around for millions of years. And in the case of papilloma viruses, they diverged or diversified as the species kind of evolved. So they infect a wide range of animal species, as you will see in some part of my talk later today. With regards to humans, there are about over 400 papilloma viruses that have been identified, 200 of which infect humans, and 40 of these are transmitted sexually. The global point prevalence of human papillomavirus infection is about 11 to 12%. And over 80% of the population will experience anogenital human papillomavirus infection at some point in their lifetime. In fact, in some populations, this is probably as high as 100%. So everybody, at least at some point in their lifetime, comes into contact with HPV. There is a linear relationship between the HPV prevalence and age, which starts after sexual debut and rises up till the age of 30 to 35, after which it remains steady for a while and then undergoes a decline in the older ages. This pattern of HPV prevalence has been seen in almost all parts of the world. The sexually transmitted human papillomaviruses are classified into low or high risk types, depending on their potential for causing cancer. There's some debate about the actual number of high risk types, but I think consensus hovers around 15 high risk HPV types. And some of these 8 high risk HPV types are known to be highly carcinogenic, while others have lesser carcinogenicity. It is persistence of the infection by any of these high-risk types that leads to cervical cancer and other anogenital cancers. There are low-risk HPV types that also infect humans, and they're most commonly associated with anogenital warts. And some infect the pharynx and the vocal cords, causing papillomatosis. In Nigeria, 17% of all cancers in women and 2% of all cancers in men are associated with HPV. The combined incidence of HPV attributable cancers range from 31 to 31.7 per 100,000. So this is a significant cancer problem for the country. So our lab focuses on the molecular biology of cervical carcinogenesis, in particular, the development of cervical cancer from the prevalent high-risk HPV infection period all through to the different stages of cervical intraepithelial neoplasia until the establishment of invasive cervical cancer. So in order to study this in our population with funding from the NIH, we established the African Collaborative Center for Genomic and Microbiome Research and enrolled 10,000 was our plan. We ended up enrolling 11,700. HIV negative by self-report, women aged 18 years and above. And we were seeing these women every 6 months for 2 years during which time they answered interview-administered questionnaire. We measured the pH of their vagina, and we obtained samples for HPV typing and blood for germline DNA in order to study genomic risks. Today, I'll be presenting results from a part of the study that we conducted in this large cohort, which focused on the vaginal microbiome in a subset of the study population. And I will explain how we obtained that subset in a minute. The cohort, these women in the ACME cohort were aged a mean of 38 years with standard deviation of 10 years. All of them have had sexual experience. All of them have had vaginal sex. A few, 15% reported oral sex, and 2% reported having had anal sex. The frequency of sexual activity in these women is shown here. Most had sex at least once a week, and others in the range of at least once a month. This is important because of the method of transmission of the human papillomavirus. So based on the HPV status of the participants in the ACME study, at the end of 2 years follow-up, we selected those participants for whom we have HPV genotyping for all the periods of follow-up to do our metagenomic study. So we enrolled 86 participants into this study. In addition, we enrolled participants who, when they presented to our study sites for cervical cancer screening, were diagnosed to have invasive cervical cancer. So in this subset of the population, the mean age of the participants was 42.9 with standard deviation of 11.7 years. 11% of them were widowed, 7% single, 1 person was divorced, and the remainder were married. So to break down these participants into the categories that we were interested in, we looked at the HPV results of the 86 participants, and we included 4 technical replicates in metagenomic samples. So we looked at the HPV status at baseline, at 12 months, and at 24 months. So this table shows N meaning negative, while P means positive. So the first row here shows women who are negative at all the times that were sampled. That's baseline, 12 months, and 24 months. And we have women who are negative twice and then became positive. This category of women we considered recently HPV infected. And women who were negative and then became positive, and the positivity persisted at 12 months and at 24 months. Then we had women who were positive throughout the follow-up period. Then we had women who were positive at baseline and at 12 months, then became negative at 24 months, suggesting that they may have controlled or cleared the infection. And then we had women who were positive at baseline only. In this study sample, there was only 1 woman, positive at baseline only, and then negative at the 2 follow-up periods. Then we had the women with cervical cancer, We had one woman with invasive papillary adenocarcinoma, and we had 2 cases that turned out after a biopsy to be non-cancerous conditions of the cervix. In one case, the patient had chronic cervicitis, and in the other case, the patient had CIN1. So this is the breakdown of the HPV test in all these participants. As you can see, there are multiple instances where participants had multiple HPV types infecting them. So this category are the women who were persistently positive throughout the study period. Excuse me, something moved. Okay. And these were the PPN, positive, positive, negative, and so on and so forth. And these were the NNP, that's negative, negative, then became positive. And you can see the HPV types that infected these women all the way to the positive, positive, then became negative. So we took the mid-vaginal samples of these participants and used it for our metagenomics experiment. So this slide just shows the pipeline of the metagenomics experiment. I will not describe that any further. So after the sequencing was done, we started our data analysis by doing an initial quality control step. And then we use Metaflan3 and Chocoflan and the Chocoflan marker gene database for taxonomic profiling in order to identify the bacteria, viruses, and other life forms that are present. So the biobakery need data workflow enables us to remove the human host reads from the sequencing data, leaving only the microbiome data for analysis. Then we mapped the sequencing results to reference genomes. In case of HPV, we mapped each sample against a database containing 461 complete HPV genomes. And for each sample, we selected the genome with the highest overall coverage and used its mapping statistic to provide a coverage histogram. I'll show examples of that. Then for Lactobacillus, all samples were mapped against a database of 45 Lactobacillus species, representative genomes. And again, the species with the highest breadth of coverage were selected to produce the coverage histogram. Then we looked at the presence of pathogenic species, and we again performed read mapping against their reference genomes. More on that also later. So we used the recommended workflow in Human3 for functional profiling of the microbial community present in each sample. And for each sample, Human3 produced tables describing the pathway abundances and coverage, and the gene family abundance table, which we then normalized to obtain relative abundances of the microbial genomes present in the sample. Well, we use Botai2 to perform nucleotide alignment and Diamond to perform translated protein alignment. So in this presentation, I will be presenting the results comparing only the participants who were negative for HPV throughout the study period. That's at baseline, at 12 months, and at 24 months. And I'll be comparing their results with those women who were positive for HPV at baseline, 12 months, and 24 months. So these are the people who have persistent high-risk HPV infection, which is the necessary cause for cervical cancer. So what we see here is the composite cladogram of all the samples. And what this shows is that we identified Lactobacillus species, which is the primary colonizing bacteria of the healthy vaginal flora. But we also found Prevotella, Gardnerella, and Mycoplasma species. Unexpectedly, we found several types of human papilloma, of papilloma viruses. Okay, so this cladogram is typical of what one expects from a metagenomic analysis of samples from the vagina. Now, at the genus level abundance map for the women who were negative for HPV throughout the study period, again, we see a microbiome pattern that's typical of healthy human vagina. In this, we see that the most abundant bacterial species is Lactobacillus, which, let's see if I can find my pointer. Okay, there we go. So here we have Lactobacillus, and that, if you use this legend, which indicates the relative abundance of the different bacteria, with the maroon indicating very high abundance and blue indicating low abundance, you will see that with all the samples present except 2, there was high abundance of Lactobacillus. That's what we expect in the healthy human vagina. But a few samples have high levels of other components of the vaginal microbiome. For example, Gardnerella, which you see here. You see some samples showing high abundance, and Adebamowo, which you see here with some of the samples. And in those cases with high Adebamowo, like this 3, 4, and Gardnerella, this suggests that those women have bacterial vaginosis. We detected a high abundance of alpha papillomavirus in one of these samples, and that's this one. And considering that all of these women are supposed to be HPV negative, this may be an instance where this participant became infected with HPV in between the last HPV typing and the sampling for metagenomic analysis. Then 2 of the samples, as you will appreciate, are not aligned with the rest of the samples. The first is this one, which shows a wide variety of bacteria present. And so bacteria like Prevotella, Adebamowo, Gardnerella were all present in high relative abundance in this participant. In the second participant, you also have a similar presence of a large number of bacteria, including pathogenic types. So this patient, this participant with the high prevalence of other pathogenic types in this particular instance also has low levels of Lactobacillus. So what we conclude from here is that these 2 participants may actually have ongoing vaginal infections or bacterial vaginosis. And if you exclude these 2 participants, you will notice that the remaining participants, so if you look at this Somebody's raised their hand. I think I'll continue and I will answer questions at the end, if that's okay. So if you look at this slides and kind of divide it into two and focus on the left side, the pattern here is clearer and consistent with what you should see in women without bacterial vaginosis with healthy vagina. So here is the cladogram for these participants and shows the different bacteria genera in different colors, and the nodes indicate the different species of bacteria present in the vagina. Now we contrast that with the microbial pattern of women who have persistent HPV infection. Lactobacillus still remains the most abundant genus in these women, but they also have a greater distribution of different types of bacteria present in the vagina. Gardnerella was the most frequent, and you can see Gardnerella here. You can see that it occurs in relatively high abundance in many of the participants. So it was detected in 10 participants on this slide, followed by Prevotella and Atopobium, Ureaplasma, Snathia, and Streptococcus. So these bacteria have the potential to cause bacterial vaginosis, and observing any one of them in high abundance or higher abundance than Lactobacillus is suggestive of vaginal dysbiosis. So this is the cladogram of the microbial pattern of the women with persistent high-risk HPV infection. All the samples in these women also have high amounts of cyprinivirus, of which I will speak a little bit more later. So I've put the two heat maps side by side for ease of comparison, and I've excluded from the women who were negative for HPV at all 3 testing periods. I've kind of obstructed those women that look like aberrant results in order to enhance our ability to compare the vaginal microbiome pattern, which shows better on this left side, with the vaginal microbiome pattern of women who have persistent high-risk HPV infection. So you can see clearly that the women who have persistent high-risk HPV infection have quite a lot of other pathogenic bacteria present in the vagina. You will also see here that there is a relative abundance of cyprinivirus, which I alluded to earlier and will speak more to in the next few slides. And this is the cladogram. So comparing the women with who did not have HPV infection at all with the women who have persistent high-risk HPV infection, you see that the women with persistent high risk have greater heterogeneity of their vaginal microbiome. So there's a lot more diversity of species because of all the pathogenic bacteria that are present. Now we look at those women with cervical cancer, and the first thing we notice is there was little or no lactobacillus present in the vagina of this woman. And that's really unusual because lactobacillus is the typical, resident of the vagina. So its presence, its absence in association with the presence of cervical cancer is interesting. The bacterial genus that were most commonly present in this category of women is Cutibacterium. Now, Cutibacterium is a marker for vaginal microbiome. So it just indicates that the sample you're studying is most probably from the vagina. But we also saw other genera like Prevotella and Bacteroides, but they were present in low abundance, except in one patient who had a high abundance of this bacteria. Expectedly, the alpha papilloma virus or viruses were present in very high abundance. As you can see, it's the last row on this heat map. So it's present in very high abundance in almost all the samples, except about 2 or 3, where it was in relative low abundance. And again, you see cyprinivirus present in high abundance. And this is the cladogram of the women with cervical cancer. It shows the marked absence of the Lactobacillus species. And here, where there is no colored image, is indicative of the fact that there are bacteria there, but they are in an unclassified strata. So we know there is bacterial genome, viral genome, and other microorganisms, but we don't know what type of microorganisms they are because of challenges with the reference database. Now, so cyprinivirus. So we were quite intrigued by finding relatively high amounts of cyprinivirus in all the PPP, that's persistent high-risk HPV infection and cervical cancer samples. This is the first report of this virus infecting humans. It's a type of papillomavirus that typically infects fish, particularly the common carp, and in whom it causes a very contagious viral disease. But we're going to be conservative in our interpretation of this result because the taxonomic classification upon which these identifications rely may be a little bit unreliable. And so we will need to continue to further analyze our data in order to ascertain the importance of this finding. So we're going to be conservative and consider that this might be a spurious hit until we have access to even more reference databases in order to evaluate it. So here we're showing the coverage histogram. And what I'm showing here is the coverage histogram of samples where we saw the cypriniviruses. And I want you to compare that with the coverage histogram of the samples where we saw HPV. So you can see visually that the blocks here are bigger, they're more solid, consistent. And so that suggests high depth and wide coverage of the genome sequences that were identified in the samples. In contrast, the cyprinid virus coverage is shallow and lacking in breadth. So that finding again suggests that we should be quite cautious in how we interpret this data, as it may be spurious, may be a reflection of contamination, which is possible or deficiencies in the reference database. Now, for the HPV, I indicated earlier that we mapped the HPV types to a database containing 461 HPV genomes. So we found several high-risk HPV types with varying breadths and depths of coverage, as I showed in the previous slide. HPV 16 and 18 are the commonest genotypes identified in the cervical cancer cases. But surprisingly, in the metagenomic data, HPV 82 was the most prevalent high-risk genotype that we found, even though a recent global meta-analysis suggested that this is a rare genotype. A recent report from Oman in the Middle East also says this HPV 82 is the commonest or the most dominant HPV genotype found in screened populations. In addition, we found the high prevalence of HPV 21. Now, this again is unusual. HPV 21 is not one of the HPV types commonly associated with mucosal surface infection. It's typically found in association with epidermodysplasia verruciformis. But we analyzed this further, and the coverage pattern we saw was remarkably consistent across all of the samples, with the same approximately 55% of the genome being covered at low depths in all samples where we identified HPV 21. So this low depth of coverage in all of the samples and the fact that we saw the same region across all the samples, again, leads us to a more conservative conclusion that this is probably an artifact arising from poor reference genome database. And during the time that we're analyzing this data, the HPV 21 reference genome in the RefSeq database was actually removed because other anomalies were detected in the database. So we could not pursue this further. We expect that the reference database will be rebuilt and be available, and then we can explore the value of this finding in future analysis. So this is what the HPV types that we found in the metagenomic data look like. And you can compare this with the results from the HPV typing using dia-LIPA, which I showed earlier. And you will notice that there are some important differences. Of course, 16 and 18 is the commonest in the cervical cancer cases, but you see this breadth, you see the HPV 21 that I just discussed, and you see HPV 82. In contrast to some of our earlier reports based on dia and linear array methods of identifying HPV, the distribution of HPV types is different. We tended to see more HPV 52 than we are seeing here in the metagenomic data. Then we looked at the Lactobacillus species, and I've mentioned the remarkable, relatively low to no Lactobacillus in the cervical cancer cases, which is quite intriguing. And because we saw all of these lactobacilli at very high depth of coverage, we believe that we correctly mapped them to the species level. So we saw Lactobacillus iners in many of the participants. Crispatus was the next, and then Jensenii and Delbrueckii, a few Gasseri, and then there were many Lactobacilli that we could not identify the species. So this is to show again this is the genome histogram showing the depth and the breadth of coverage of the sequencing and how we were able to map the Lactobacillus to the reference database. Then we looked at the pathogenic species that may be present. And for Gardnerella vaginalis, wherever it is present, we're able to identify it in high abundance. So this genome histogram shows Gardnerella. So you can see that we had quite a high depth and breadth of coverage of the genome. Similarly, for Atopobium vaginae, again, we have very good coverage, and this species was significantly commoner in the women with persistent HPV infection compared to women without persistent high-risk HPV infection. Then we saw an unusual bacteria, the Bacillus indolicus, in 4 of the women with persistent high-risk HPV infection. This bacteria has only just been recently isolated and identified in the female genital tract. So we did a little bit more mapping to its reference genome. So you can see our genome coverage histogram is E and F, the last 2 here. And you can see again that despite the occasional peaks of high coverage, only a small fraction of the genome of the bacteria was present in the samples. So this again requires further studies to look at the potential role of this newly discovered bacteria in as a cofactor of persistence in high-risk HPV infections. So I'll round up by talking about the functional profiles of the bacteria that we identified. So the mere presence of the bacteria is one thing. Their activities, of course, is what we're most interested in. So we looked at the pathways that are enriched in the different groups that we're comparing. We looked at the top 5 pathways that were enriched in women without HPV infection. And I won't go through each of the names or what they do, but these are the pathways that are enriched in women without HPV infection. And this part, excuse me, the biggest contributors to these pathways are the Lactobacillus species and bacteria in the unclassified strata. So quite a lot to unpack in terms of what is going on with the pathways. In the women with persistent high-risk HPV infection, you will notice that the top pathways are completely different. So this gives us some hints about the functional activities of the bacteria of the vaginal microbiome in women with persistent high-risk HPV infection. And most of these pathways are contributed by Lactobacillus species, Gardnerella vaginalis, and bacteria in the unclassified strata. Then we looked at the gene families, and in the gene families, again, we see there's a difference in the top enriched gene families comparing women without HPV infection with women with HPV infection. So we conclude that our taxonomic classification identified a higher diversity of potentially pathogenic bacteria in women with persistent high-risk HPV infection. and that this could mean that persistent high-risk HPV infection leads to imbalances in the flora of the lower female genitalia and the reduced prevalence of lactobacillus, which could predispose women to development of bacterial vaginosis. The analysis of cervical cancer cases showed remarkable absence of lactobacillus species and decreased bacterial diversity. We also highlighted the discordance between our metadata and the metagenomic data, which highlights the ongoing challenges with the reference genome databases for the bacteria and viruses that we encountered in this study. So improvements in those reference databases will help us to better characterize the vaginal metagenome and identifying potential therapeutic targets for controlling persistent high-risk HPV infection. So this work would be impossible without the staff who worked diligently on this project over the period of several years. And some of them are in this picture, many are not. And the investigators who collaborated per University of Maryland conflict of interest rules, I have to mention that one of the investigators is my wife, Sally. But all the sites, had site investigators who contributed tremendously to the study. These are the sites in Nigeria, and of course, institution in the US and Geneva in Finland that provided bioinformatics support. So this presentation has been the work that I described, supported by the H3Africa U54 grant, and some of the foundational work was supported by the CADA grant from the NCI. I should also mention that we continue to receive support from the University of Maryland Cancer Center Support Grant and the State of Maryland Tobacco Restitution funding. Thank you very much.
Thank you very much, Prof. Clement, for such a fascinating and very insightful talk. We have a few questions. I think I'll just get to it very quickly so we can cover as much as possible. The first question is from Oscar, and his question is, thanks for a great presentation. Did you assess for effect of HPV vaccination on HPV risk? That's the first one. And also the effect of vaccination on metagenomic diversity.
Thank you. So HPV vaccination is not yet common in many sub-Saharan African countries. I think except maybe Rwanda and maybe South Africa, a significant proportion of most African countries have not started to systematically vaccinate their population. So none of the participants in this study have received HPV vaccination.
Okay, thanks, Prof. I think the other question was if the vaccination affected metagenomic diversity.
Yes, so since in this study, no one has received a vaccination, we cannot answer that. But okay, on the global level, no one has, as far as I know, studied the impact of HPV vaccination on the vaginal metagenome. There are, very few studies of the metagenome to start with, the vaginal metagenome to start with.
Okay, excellent, thank you. You can also please put on your video if that's okay with you, sir, so we can actually see
Yeah,
your
so
face.
I'm trying to do that. It says on my end that, that the video is on, but I can't see myself either.
Okay, no, that's fine, that's fine. I just answer the next question. There's a question from Joanne Passmore. She says, thank you for this important analysis. I wanted to ask whether you consider that bacterial vaginosis increased HPV risk and persistence due to BV disturbing the mucosa and allow better access for HPV to the basal membrane.
Not in this study. So one will need to, you need a different method to answer that question. I don't think the metagenomic approach is the ideal way to answer that question. You need spatial metagenomics and transcriptomic method to address that question.
Thank you very much. There's another question from Imran. And the question is, could these metagenomic data explain the concept of HPV latency period vis-a-vis reinfection and clearance?
That's a very interesting question. My suspicion is that our data, in fact, throws even more controversy into that question about what does it really mean when you say that a person has cleared their HPV, because the ability of the different tests to identify the presence of HPV varies. Most commercially available approved screening tests have specific settings for the number of HPV virions present when the test will become positive. So we call those, diagnostic tests. In this study for the HPV genotyping, we used an epidemiological test, which is the dia-LIPA, which is sensitive to as low as 10 virion particles. With metagenomic sequencing, depending on depth of coverage, you can be relatively confident. If the metagenomic analysis says there's no HPV, then there's no HPV. So it does kind of creates new questions in terms of clearance, reinfection, and things like that. But it doesn't answer them. It generates new questions, but it doesn't answer them.
Wow, interesting. So there's another question from James. And is there any potential role for the use of lactobacilli to reduce chances of vaginal cancers?
Yes, and not just lactobacillus. So you have to consider where the lactobacillus that infects the vaginal canal originally comes from. Okay, so it has to be from the anus. It has to be from the gut. Okay, so there is potential that by giving people lactobacillus orally or by other methods, you may change the characteristics of the vaginal microenvironment such that it becomes less prone to sustaining a persistent high-risk HPV infection. So there are some studies that have done that, and the results, I think, currently are interesting, and some, you probably hear more about this soon from pharmaceutical companies and food supplement companies promoting methods of changing the vaginal microbiome to make it healthier, quote unquote, reduce vaginal dysbiosis, and reduce risk of infections. So there's been a lot of work in that area.
Wow, quite a bit to unpack still. Thank you. So there's another question from Peter. He's asking if the fact that vaccination has not been received pose some ethical concerns, and that's what are some of these ethical challenges that may begin to arise, and if you actually encountered any during the course of your research.
Well, I think HPV vaccination remains the goal for all populations, but it's expensive. And many African countries lack the delivery infrastructure. If HPV vaccines could be delivered as part of infant immunization, I think we will have seen a higher implementation rate, but it requires administration at a specific age group. Yeah, recently that age group has been extended, but you need to have a functioning adolescent healthcare system that you can use to deliver the vaccination. Otherwise, it just will be a tremendous challenge to do. So from my perspective, from our perspective as people studying HPV, it reinforces the need to continue to look for better ways of screening for HPV and for doing cervical cancer prevention, because it's still going to take some time, maybe decades even, before many African countries have the resources to roll out HPV vaccination at population level. Until that time, a significant proportion of the female population will have been exposed to HPV. And of course, once you're exposed to HPV and you're infected, then the vaccine is no longer relevant for that population. So we have this large population of women who are going to be infected, who are going to be at elevated risk of cervical cancer, for whom we need to continue to implement screening. Because right now, even with the screening technologies that we have and we know to be effective, we don't have high population coverage. So we need to discover even newer, less complicated modalities, which is part of what we do at the ACME Research Group.
Thank you very much, sir. There are quite a few questions still. Time is far spent, but I'll just go through them and see if you could actually share some thoughts on them. Okay, the first one is just for you to comment about sample size determination in metagenomic studies. Very, very briefly, please.
That's a good question. So, so the easiest answer is sample size determination in metagenomics is based on funding, not biostatistical concepts, because metagenomic sequencing is still extremely expensive, and so that limits the number of samples that you can sequence. We, of course, expect over time that just like other omics technologies, the price will fall. So we have thousands of mid-vaginal samples from these women that we will be happy to sequence at that time. So right now, we do largely just the kind of comparisons that I showed, and then one can categorize the samples on the basis of the presence of vaginal dysbiosis or no dysbiosis, or you can examine specific components of the microbiome, which we have done, for example, in looking at mycoplasma and its association with persistent high-risk HPV. And when you narrow down like that, then you can do sample size calculations and you can do follow-up studies using simpler omics technologies, which are relatively cheaper. But at the moment when we're really just trying to look at what is the nature of the global metagenomic environment, you have to do this large sequencing experiments.
Oh, excellent. I think we'll have time for 2 more questions if possible, because it's already on the hour, from Prof. Nikki Mulder and from Chantal Klassen. So the first question is, your metagenomics analysis focused on the bacteria and HPV. Did you look at possible fungal components of the microbiome and their potential impact? That's the first one from Prof. Mulder. And there's another one, but I'll let you just quickly give some thoughts there.
Yes, we did look at fungi, and we didn't see anything interesting going on. I should mention with some of the things I kind of took out in the interest of time. It's important in order to be able to characterize what is really the, shall we say, physiological vaginal microenvironment from the vaginal microenvironment of individuals who may have ongoing STDs. So one of the things we did for all our participants is to do a screening microscopic culture and sensitivity test. And if you're positive, then we treat you for that and we don't enroll you in the metagenomic study, because all that will show us is one of the important things that will show us is the presence of those STDs. So we excluded many of those potential participants. And I guess when we write out the paper or in other presentations, we will show that there is actually a very high prevalence of asymptomatic STD in this population, and its potential role as a cofactor for persistence of high-risk HPV infection and cervical carcinogenesis.
Excellent. Thank you so much, sir. Just one last question, we can just squeeze that in.
Sure.
Okay, from Chantal Klassen, it says, why do you think you're seeing different profiles in the NNN group? Do these women have any clinical characteristics which could explain this? Also, were any of the cervical cancer patients associated with HPV groups?
Why are we seeing different vaginal microbiome profiles in the NNN? So what we saw in the NNN group, which are women without HPV infection, is typical of a healthy vaginal flora. So it's not unexpected. Then in the cervical cancer, we did see different HPV types, and I showed a slide showing the types of HPV present in the cervical cancer group, which predominantly type 16 and 18.
Thank you. Thank you so very much, sir. Thank you for this amazing session. Thanks for your time. And thanks to everyone for the questions and for attending. And we're looking forward to further output from this research. There's lots of compliments, comments, and just appreciation for the work you're doing on the chat, if you get a chance to read it. And also just to say that to everyone that attended, we do have a task force in H3Africa, Microbiome task force, and we do meet on every 3rd Monday of the month at 4 p.m. Central African time. And if this is something of interest to you, please feel free to get in touch with the coordinating center and they will direct you accordingly. Thanks so much again, Prof. We really appreciate your time here and for this very, very amazing work.
Thank you for giving me the opportunity to share the work of not only myself, but a large number of people that I've tried to represent today. Thank you very much.
Thank you, sir. Bye, everyone. Have a lovely evening, morning, wherever you're at. Thanks. Bye-bye.